Hi everyone. Thank you for attending today's webinar. We are going to share with you a presentation, and just after we are going to make a quick Q&A session with our technical specialist, Cédric Sapé. So if you have any question during the presentation, do not hesitate to ask them, and we will try our best to answer them at the end. Enjoy the presentation and see you just after.
AAV Biosciences is a French biotech company founded 20 years ago, specializing in molecular delivery systems, present in France and in the US. The French site is located in Marseille and comprises the headquarters and the production site. It's divided into 2 laboratories and is responsible for R&D, production, the mRNA and LNP platform, marketing, and sales. The US site is in San Diego and deals with marketing, sales, and business development. Over the years, we have settled a worldwide distributors network. One of our objectives is to develop the spread of our reagents and expertise. This expertise is based on more than 20 years of experience in innovation. Several important milestones have marked the company's history, such as the commercialization of the first magnetofection reagents in 2005, the creation of OZB Inc. in 2013, and our recent acquisition of new facilities for development of LNP and mRNA platforms. We're proud to cover a huge variety of fields from bioproduction to viral applications, going through in vivo, protein delivery, or stem cell applications. All the products developed and sold, as well as our platforms, are under the ISO 9001 certification. These innovative reagents allow OZ Biosciences to offer solutions in 3 main areas: cell and gene therapy, bioproduction, and custom platform productions. Regarding genetic modification of cells, several methods can be considered, but even if they each present some interest, they are not optimal for cell engineering. Electroporation or nucleofection are very efficient transfection methods with a rapid and high transgene expression, but they induce high toxicity and require expensive material and specialized equipment. Lipofection and polyfection are often used because they are simple and quick transfection procedures that require low optimization. They show some reproducibility, but they present high yield of toxicity with a low to moderate efficiency. Finally, viral infection is maybe the most efficient way to genetically modify cells. However, it's technically challenging for the creation of viral vectors. For example, they can be very expensive. And most of all, viral particles may provoke immune reaction, impairing final results. In order to increase the viral vector capacity to genetically modify cells, there are 2 possibilities: physical or chemical solutions. Concerning physical methods, we have developed the magnetofection technology, which consists of magnetic complexes that are attracted by a magnetic field and clustered onto the cell's surface to favor viral entry. It's efficient, non-toxic, and adaptable, but it does require additional steps. Spinoculation is another way, which consists of long centrifugation. It's cheap, easy, and non-toxic, but not very adaptable nor very efficient, and requires additional steps. In parallel, there are chemical methods, such as the use of polypeptides like retronectin or vectofusion, that are efficient and non-toxic, but quite expensive, hard to handle, and not adaptive to every cell culture model. Cationic polymers like polybrine are well known and often used. They're cheap and easy to handle, but toxic and not very adaptable nor efficient. And finally, polymers like LentiBoost that need additional cationic compound to be efficient. They are non-toxic, very efficient, and cheap, but not very adaptable. In response to users' needs, we have developed and patented a new class of modified cationic polysomers. They are non-toxic, versatile, and highly efficient. They take up the positive points described above while eliminating as far as possible the negative ones. Despite their differences in formulation, AAVBlast and LentiBlast have similar characteristics and functions. They play at various levels to increase infection and transduction. Due to their charge, they neutralize electrostatic repulsions between membrane and viral particles, favoring the attachment of viral particles to the cell even in absence of a receptor. Their integration in the cellular membrane maximizes the permeabilization. They thus promote the fusion of the viral envelope with the cell membrane, improving the entry of viral particles into the cell. In the case of retroviral vectors, they are described to play a stabilization role during reverse transcription and DNA trafficking to the nucleus. To address AAV viral particles that are promising candidates for gene therapy, we have developed AAVBlast, a non-toxic, efficient, and versatile viral enhancer. AAVBlast is non-toxic, as demonstrated here on the Vero cells, where increasing doses of AAVBlast do not change the proliferative index of the cells. It works also for primary cells, such as bovine mesenchymal stem cells, where the survival rate indicates that not only the viability is not affected, but also it induces proliferation. An interesting feature of this polysamer is that it acts as a thermoresponsive gel, and so it can stabilize and protect AAV up to 30 days at 4 degrees Celsius compared to non-treated AAV. As we see here, after 30 days, the AAV serotype 2 mixed with AAV-BLAST are still active. So of course, AAV-BLAST enhances the transduction efficiency of AAV of various serotypes in cell lines and primary cells. As an example, we can cite the work by Misoki that demonstrated an increase of transduction in HeLa and SAOS2 cell lines and HUVEC primary cells transduced with EGFP, AAV2, and AAV-BLAST in a dose-dependent manner. They also validate AAV-BLAST action to increase transduction both in terms of percentage of infected cells and fluorescence intensity with AAV from various serotypes 2, 6, and 8 in primary ovine mesenchymal stem cells, demonstrating the versatility of the reagent. As the authors stated, the experiments revealed that AAV-BLAST not only increased transduction efficiency in a dose-dependent manner, but also promoted cell survival. Enhancement was seen with AAV2, AAV6, and AAV8 capsids, confirming the three main characteristics of AAV-BLAST. For retroviruses and lentiviruses, we've developed LentiBlast Premium. Its formulation is non-toxic, efficient, and money-saving. The lentiviral vector field of action spans from in vitro applications to gene therapy, meaning that a high MOI is often required. A high MOI means high volume of viral production with long and costly purification and concentration steps. Consequently, high volumes of production mean huge amounts of plasmids encoding viral particles and large volumes of transfection reagents to transfect extensive cell cultures. With the viral dose increases the numerous hazardous and costly steps from transfection to production. So what if for the same transduction efficiency we could use 10 times less viral dose? This would mean that the load of virus would be lowered, the cost will decrease, as well as the immune response. And this is what LentiBlast Premium offers, since it allows to reach the same efficiency as virus alone at a 10 times superior MOI. As we can see here, LentiBlast Premium added to 0.025 MOI induces the same efficiency as non-treated cells infected with 0.25 MOI. The same for MOI 0.25 and 2.5. The most important feature of LentiBlast Premium is its capacity to increase lentiviral transduction, as demonstrated here in hematopoietic stem cell line KG1A and primary CD34 plus cells. The stem cells were infected with GFP encoding lentivirus using a MOI of 5. As we can see, LentiBlast Premium dramatically enhances percent of transduction in both cell lines and primary cells. And if we have a look at primary CD34 plus cells, we can see that not only the percentage of GFP plus cells is increased, but also the overall protein production. We compared our reagent to what was already on the market to make sure it responds to users' needs. In this experiment, KG1A were infected with GFP encoding lentivirus at 3 MOI in presence, or not of principal commercial transduction enhancers. The percentage of GFP cells was measured 72 hours after transduction by flow cytometry, and fold increase in transduction was calculated in comparison to non-treated cells. LentiBlast Premium reached its highest percentage of GFP positive cells compared to competitors, and looking at the fold increase demonstrates that it works great even at low MOI. When comparing LentiBlast Premium to the most well-known chemical polymer-based transduction enhancer, polybrine, we can see that even when used 100 times the recommended dose, the cell monolayer is not affected. Neither is the cell proliferation rate, demonstrating that LentiBlast Premium is non-toxic for the cells. Thanks to its principal features, efficiency, non-toxicity, and cost-effectiveness, LentiBlast Premium was used in numerous cell types, such as primary myeloid precursors, trabecular meshwork cells, or cardiomyocytes, allowing authors to publish articles in high-ranked journals. Now let's see some examples of what LentiBlast Premium allowed to accomplish. In this first example, primary acute myeloid leukemia cells from patients were transduced with virus in presence of LentiBlast Premium. As stated by the authors, the transduction efficiency reached 30 to 60% and allowed them to understand how mutations in RNA splicing factor impact cellular functions in 2 models of pathologies. In the next example, LentiBlast Premium was used to improve the ex vivo transduction of mouse hematopoietic stem cells with a SUMF1 lentiviral vector. The genetically modified hematopoietic stem cells were then transplanted into recipient mice to correct the lack of SUMF1, thus fixing up biochemical deficits and improving neurocognitive function in deficient mice. In Burton's study, LentiBlast Premium was used to enhance the transduction of hematopoietic stem cells with a retrovirus. The transduced cells were transplanted into lethally irradiated mice. LentiBlast Premium facilitated the efficient introduction and tracking of specific retrogenic TCRs in cells. In short correspondence with the author, they told us they have used LentiBlast Premium and that it certainly helped them in accomplishing some difficult experiments. Finally, in this last example, Stamaria's paper uses LentiBlast Premium to facilitate the transduction of primary human T cells with a lentivirus encoding a chimeric antigen receptor. By evaluating transduction efficiency at day 7, the authors counted more than 96% of CAR-positive cells. These CAR T cells express genetically modified receptors that recognize and bind specifically to tumor antigens, triggering a cascade of tumor cell destruction. It is true that even if our modified polysomers were developed and formulated to give the best efficiency, in some cases there may be a need for optimization. In this way, it is possible possible to optimize the volume of enhancers. Several concentrations can be tested from 1 to 1000 to 1 to 25. This is often recommended when MOI is unknown. We have also observed that some cell types get better transduced if the complexes of viral particles and reagent are added before the cells. They get transduced while they settle and colonize the cell plate. It is also possible to play with the serum part of the medium by varying the concentration, for example. And we have to keep in mind that depending on the viral particles, some infections work better in medium without serum. Finally, it is also possible to replace the transduction medium after 24 hours for the cells to be in their optimal culture condition. All these tips would apply to both enhancers. Okay, so what do you need to get started? The only requirement is either LentiBlast Premium or AAVBlast reagent that are composed of one tube and your viral vector. That's all. And it's compatible with any cell culture dish from culture plate to culture bags. In parallel to LentiBlast Premium, we have developed LentiBlast Premium Superior Grade. It's identical to LentiBlast Premium, but with advanced quality controls, such as chemical characterization or advanced purity assessment. LentiBlast Premium Superior Grade meets the USP 63 and 85 criteria respectively for mycoplasma and endotoxin dosages. It is now designed preferentially for use in preclinical and early phase clinical trials and can be used as an ancillary material for cell and gene therapy. We're also actively working on the development of LentiBlast Premium GMP grade that should be available soon. This grade of production put LentiBlast at the first place to integrate cell therapy workflow. This graph illustrates how LentiBlast Premium can incorporate the cell therapy pathway following stem cell purification and their ex vivo activation. Its capacity to increase gene transfer and overall efficiency while reducing time, cost, and viral dose fits perfectly during major steps of hematopoietic stem cell modification process. LentiBlast Premium would enable the enhancement of retroviral or lentiviral mediated gene transfer prior to the transplantation of genetically modified cells. So what I would like to emphasize before ending this webinar is the fact that since it's been launched, LentiBlast Premium has been cited by about 100 high-ranked journals. This success is due to the fact that LentiBlast Premium and AAVBlast are non-toxic and allow to increase viral infection and transduction on any cell type and culture model. They lower MOI, time, and cost of experiments. This makes the modified polysomers ideal candidates for gene and cell therapy applications. LentiBlast Premium and AAVBlast are part of a complete range of reagents for viral applications. They belong to the infection enhancement section associated to magnetofection tools such as Viromag-RL or Adenomag. We also have developed reagents and kits for viral production in HEK293 T cells such as HYVIR or capture concentration and preservation such as Mag4C line of reagents.
So thank you everyone for your attention. We are now going to go for the Q&A session. If you have any question following this webinar, you can contact us at contact@aussiebioscience.com. And for any technical question, you can contact us at tech@aussiebioscience.com. So the first question will be, you have shown that LentiBlast Premium is almost equivalent to LentiBoost and retronectin. So what is the advantage?
I will say that there are several advantages. For example, LentiBlast Premium works better at low MOI, and we have seen that the fold increase in transduction is higher than other competitor when we talk about MOI of 2 or 5. We think that at high MOI, the viral dose will compensate the effect of the enhancer, and thus the effect of LentiBlast Premium will be less visible. But we know that it is better to go with low MOI since the immune response will be lowered. So in my opinion, this is the first advantage. Then we can talk about the handling of the reagent, because the handling of LentiBlast Premium is easier. It comes as one tube, ready to use, and there is no need to prepare a cell plate or to coat culture bag, such as we have to do it with retronectin. You add directly LentiBlast Premium to your virus and it's done. So it is one tube, one condition, adaptable, ready to use. It reduces the immune response, and most of all, it is totally non-toxic. So yeah, there are plenty of advantages.
Thank you. Another question will be, did you compare the efficiency of LentiBlast Premium to magnetofection?
Yes, we have extensively compared LentiBlast Premium to magnetofection since we own also this technology. And the conclusion is that the increase in the efficiency is almost identical if we compare both of the methods. In fact, it will really depend on the cell culture and the virus type. But if we average all the experiments, the increase is quite the same. Magnetofection will be better for some cells, also virus types, and LentiBlast Premium will be better for other cell types and virus. But there are some cells, of course, such as hematopoietic stem cells, that prefer LentiBlast Premium. But in most cases, both methods will give the same results.
Okay. Another question will be, can we add LentiBlast Premium to our protocol with spinoculation?
Yeah, definitely the two methods are compatible, and then they can be combined without any problems. And moreover, this would allow to gain in efficiency. Of course, once again, it depends on the virus and the cell type. And if I remember well, we have some publications that have demonstrated that LentiBlast Premium can be added to spinoculation, so it works. And one point before going to the other question, concerning the protocols, there is no need to change the protocol. You just have to mix the virus with LentiBlast Premium and follow spinoculation process, and it's done.
Very easy.
Yeah, definitely.
Then please let me know if is it okay to use LentiBlast Premium with polybrain when I transduce the cells, and if not, can you explain why is that so?
Yeah, no, not at all. We do not recommend adding polybrain to LentiBlast Premium, such as for other poloxamer. There is no gain at all of doing this, as opposed to what we can see with other physical methods, such as spinoculation or magnetofection. Chemically speaking, polybrain is a cationic polymer, and LentiBlast Premium is also positively charged. So there is no need to add positive charge to something that is already charged. And moreover, the main reason will be that the index viability will be dramatically damaged with the use of polybrain in association to LentiBlast. LentiBlast Premium is totally non-toxic and does not impair differentiation of the cells, where polybrain is known to be highly toxic. And this becomes dramatic if we increase the dose to gain a better efficiency. So no, it will not be a good idea at all to combine polybrain with LentiBlast Premium.
Okay. And yeah, this will be our last question for today. So how can you explain that AAVBlast protects AAV particles? Is it due to the fact that it contains other substance from animal origin, for example?
Not at all. No. AAVBlast contains only components that are derived from FDA-approved polymers dissolved in water. There is just no substance subjected to a restriction of any kind, and it contains no substances from animal origins. Actually, it is a polymer, and it is 100% synthetic. And so we believe that the protective effect comes from the fact that AAVBlast has thermoresponsive property that enables him to form a gel when it is at 37 degrees. And so the gel will protect the viral particles and help them to reduce their degradation. And so the stability will be enhanced. And not only it will allow to permeabilize the cellular membrane, but to protect also the AAV. So they are more preserved, actually, by this gel effect.
So thank you very much, Cedric. So this is the end of today's webinar. If you have some times just after, we made a quick survey to see if you are satisfied with our webinar. So do not hesitate to answer it. And if you have any question, you can always contact us. Thank you very much.
Thank you.